All right, so in mitosis, it seems like the most difficult part of the whole story is getting those chromosomes doubled, or getting your chromatin doubled, somehow compacting that into chromosomes and making it organized so that you can split those chromosomes, as in the whole chromatid story, so that the chromatids can become eventually chromosomes. Remember that what we want to talk about here is how are we going to split the cell? Remember what we have. We now have a cell with 2 nuclei in it. And those nuclei are starting to decondense. The chromosomes are going down and they're becoming chromatin again. Your nucleolus has redeveloped. Well, how are we going to split this cell? Because eventually what we want to form from this is what we call 2 identical daughter cells. That's our eventual goal. And that sounds like quite a trick. You know, it almost makes mitosis pale by comparison. And remember where we are here in our little cell cycle. We've been through a normal cell lifespan. We're in that 10%. We've gone through what I've labeled here as M, the mitosis part of the mitotic phase, and now we're heading into cytoplasmic division. We need to divide that cytoplasm. So let's take a look at cytoplasmic division from 2 perspectives, the perspective of the animal cell and then the perspective of the plant cell. All right, how does an animal cell divide its cytoplasm? Well, here's our dilemma. Our dilemma is this. When we observe it under a microscope, we see that what happens is you go from a relatively round cell and the cell begins to elongate into almost an oval shape. And how does a cell elongate? How does a cell do that? Well, logic dictates that a cell being a fluid cell or plasma membrane, there's really no, levers. When I move an arm, for example, I'm levering it, okay? When I do this and I elongate my hands, I'm using bones and muscles as levers. Well, to a degree, there is none of that in a fluid membrane, but let's not forget something that you know about from your lessons in cell biology. And let's not forget that entire cytoskeleton. And let's not forget the fact that there are microtubules and microfilaments that connect this cell in an unbelievably intricate way. And that these microfilaments are indeed anchored across the cell. In fact, if you know your cell biology, you know of 2 microfilaments called actin and myosin, which these things really come up a lot whenever we talk about motor molecules. You know, we're going to talk, you could link to the lesson now on the muscle. And muscles work by the interaction of thin filaments and thick filaments, which kind of slide on each other. So, you know, and to make a short story long, what happens with muscles is you get this thick myosin filament that looks like that, and then a ring of actin filaments around it. Like so. And these filaments slide on each other by motor molecule anchors. And so what you can get here is the same situation here. You can actually form sliding filaments, and if you can slide filaments, guess what you can do? You can shorten those filaments. Now think of that. Can you form an oval from a round circle by sliding filaments on each other? Let's see. Well, let's picture my cell. Let's go to one side of the cell. It's round, but I have microfilaments across. Imagine I pull those microfilaments in that direction. Well, particularly at the center. Well, what's going to happen then is, wow, this thing is going to bend inward like that. And if we look at the other side and we do the same thing, remember it's round, and we bend it inward like so. And it doesn't have to necessarily be the same microfilament. This microfilament could be pulling in one direction and the one right next to it could be pulling the other direction. What's going to happen here? Well, gee whiz, the same thing is going to happen. That round membrane is going to bend in. And now let's connect these, and look what you have. You have the beginning of what I've referred to as the cleavage furrow in animal cells. And indeed, that's exactly what begins to happen. We see under electron microscope studies that microfilaments are literally pulling this thing in, and some microtubules are involved in this too, but I really want to stress these microfilaments. And so what we begin to get is an amazing thing, and let me just show you a photograph that just knocks me out. You actually get the beginning of cell bending. This is taken from a developing frog's egg. And this egg has been fertilized and has begun the process of development. And the first thing that happens in development is the beginning of that cleavage furrow, which will eventually fuse. Now, let me make sure you understand what I mean by this whole idea of fusion. Eventually, these two cells are going to fuse, and let's see how that's going to happen. Or these two parts are going to fuse. Here's the one cleavage furrow, and I'm going to make these very pronounced, and there's the other one. Okay? Now, remember what we did back in the mitosis part. We have our nucleus. Okay? The chromatin is now an amorphous. We're way past. We're in way late telophase now. Okay? And we have our nucleus and our nucleolus and the chromatin. And now look what can happen here. If we simply pinch those together, let them merge, you'll eventually go into two, what I've referred to as daughter cells. Everyone refers to them as daughter cells, to be quite honest with you. And these two daughter cells will be identical. And so in an animal cell, we see, therefore, this whole idea of the cleavage furrow, and we see this whole idea of the splitting of the cell thanks to the contraction of microfilaments. But what about animals? What about plant cells? Okay? In plant cells, we're going to have a completely different story. Why? What do plant cells have around the outside of them? Cell walls. That's a dilemma. Why is that a dilemma? Well, let's take a look. If we take a look at a plant cell wall, here's what we have. A rigid, non-flexible, non-cleavable structure. It's hard. It's got cellulose and pectins, and you're not going to bend that. But what a plant cell has is a cell membrane. But we can't do a cleavage furrow in the cell membrane. It's going to leave the wall. But what can we do? Well, here we are. We're in late telophase. We've made our nuclei. We've got our nucleoli. Okay, what can we do? Well, we can synthesize new membrane. What's the organelle that synthesizes membrane? That's right, the Golgi apparatus. So we could pop off Golgi vesicles. And remember, there's a whole bunch of these little highways called microtubules and microfilaments that Golgi vesicles might be able to migrate along. And so they could migrate toward the center of the cell from both cells and start to form a chain of Golgi vesicles. Now, that being said, what can happen to these Golgi vesicles is they can fuse with each other, and look what's going to happen. What are Golgi vesicles made out of? Membrane. And what's in those Golgi vesicles? Proteins, maybe enzymes that can actually build a new cell wall. And look what I've just formed, folks. I've just formed a new cell membrane. Let's fuse those. Let's fuse those. Two plant cells back to interphase. And then, of course, the plant cell can grow and then eventually add secondary cell wall and harden off and go through the process again. So I just want to leave you with one lesson. Don't make mitosis a mystery. Don't make cytokinesis a mystery. Learn the process. It's the basis of the way cells reproduce.